All right, so today we're going to talk about insect development, which is chapter 12. And overall, this shows for a caddisfly the different places that an insect might lay eggs. So on vegetation near the water, on the surface of the water, just below the water, dropping to the bottom, down on the substrate. You might have adults flying underwater to lay their eggs, emerging again and laying eggs elsewhere, or maybe just making one trip underwater. The eggs then, denoted by number 1, hatch into larvae, and the larvae grow through multiple instars. Eventually, for caddisflies, sometimes getting large enough to create cases for protection or create nets underwater. And then they pupate, generally in a case on the substrate. They pupate and turn into adults and emerge. Sometimes they emerge underwater. Sometimes they emerge on the surface or on emergent vegetation. And then they fly around and have their adult life cycle, mate, and lay eggs again. So in general, aquatic and terrestrial insects don't really differ much in terms of how they develop. There are EMBRYOGENESIS, HEMIMETABOLOUS, and HOLOMETABOLOUS insects. And in typical development, there's both growth, which is an increase in size and biomass, and development, which is the formation of new cell types, tissues, or organs. So embryos and larvae both grow and develop. Pupae don't usually grow, they just develop. And then adults typically do neither. They neither grow nor develop, and they're just maintaining their adult body. So to start off with, the embryo develops inside the egg, and this begins at fertilization. There's the multiplication of cells through mitosis. There's development. We have different types of tissues developing, different types of organs developing. And then eventually the young insect must break through the chorion of the egg and the vitelline membrane. They might swallow air or fluid to kind of like bulge out and create pressure on the egg surface. The egg itself might have lines of weakness or hinges that can pop open. And then some larvae have these egg bursters. So on their first instar, they might have a specific kind of, hard or pointy feature that allows them to push through the egg, like shown in the picture. So here's another picture of an egg burster. You would only see this on the first instar, and the second instar would no longer need it, and typically that would disappear. So then as the larvae grows, this is the main function of the larval stage, is growth. And so, but a lot of these organisms have exoskeletons, which keeps them from growing. So what they have to do is they grow until their exoskeleton is maxed out, and then they have to molt that old exoskeleton and grow again as a new instar. So each molt produces a new instar, and the length of time that the organism is in that instar is called the stadium, the duration of each instar. Hypermetamorphosis is when an organism has different instar morphology. So some caddisflies in the microcaddis family, Hydroptilidae, they actually look like little, almost like little beetle larvae in their first 3 or 4 instars. And then they change dramatically, and they look a lot more like caddisfly larvae, and they produce cases in their latter instars. And we noticed this at our study sites at Mount St. Helens. We found these really, really tiny little guys, and they were super hard to identify, and we couldn't really figure out what they were until someone mentioned that this difference, this hypermetamorphosis, this difference between early instars and later instars, in particular for the hydroptilid caddisflies. So at each instar, when it reaches its max, it goes through this molting process, and there's a few different stages. There's apolysis, which is the separation of the existing cuticle from the epidermis. And then there's ecdysis, which is the shedding of the old cuticle once a new one has formed. And this in-between state, when when an instar is molting and has developed a new cuticle and is shedding the old one, we call that being in kind of a pharate or a cloaked stage. So here are some of the different stages and where you can find these pharate forms. So when the egg hatches, it's growing, and then there's apolysis, this pharate stage, and then ecdysis, and then they're growing again. And then at between each instar, we have these pharate stages. And then you also have a pharate stage between the last larval stage and the pupa, and then also between the pupa and the adult. So during molt, insect larvae are very vulnerable and sensitive. We call these tenaral stages. The reason is that they have to detach their muscles from the old cuticle and reattach. So often it's really hard for them to move, or if they do move, they risk kind of deforming this growing exoskeleton. And so this, like this says, muscle action on the new cuticle before it's hardened can cause deformation. So of course that would be damaging. So molting is under both endocrine and nervous system control. And this little diagram just shows you, what happens. So like you have normally an epidermis and a cuticle. And then during apolysis, you have the separation of the epidermis from the cuticle. And then you have the secretion of fluids and the growth of the epidermis. So you have mitosis happening. You have cell division in the epidermis, which is the organism growing a new body. And then you have secretion of a new cuticle layer on top of that epidermis. And then you have activation of enzymes that help to break down the old cuticle, and then the organism can break out of the old cuticle. Okay. So when they break out of the old cuticle, they leave behind what are called exuviae, which are so beautiful and fantastic. Basically these shells of their old exoskeleton. Usually you can see where they've cracked open the back and crawled out of the back of the exoskeleton and left these behind on rocks or on plant pieces. And we have a stonefly, a mayfly, and an odonata zygopter, an anisoptera. I think, which one is that? It's hard to see. Yeah, that's an anisoptera, so a dragonfly. So ECDYSIS, there are these proteinases and chitinases. These are both enzymes. Anything ending in ase is an enzyme. So proteinase is something that kind of breaks down proteins, and chitinases are things that break down chitin, which is what the exoskeleton is made of. And so these things help to digest the old cuticle. The insects swallow water or air to split the old cuticle and to kind of release themselves. And then there's a series of peristaltic waves of contraction or wriggling to kind of get out. The new cuticle then is allowed to kind of stretch, and they might take on more air or water to get that cuticle to stretch before it hardens. And then it hardens, it becomes sclerotized or tanned, it turns dark brown. So here you can see a cicada crawling out of its old shell, and it's this lovely green color. The old shell is this brown color, the hardened tanned color, and eventually the cicada will harden and tan that brownish color, but when it first emerges, it's this beautiful green. So different species go through different numbers of instars, from as few as 30 up to 50. And we think many instars, like 50, is the plesiomorphic condition, and that over time, insects have evolved to needing fewer instars. The variation in instar number, though, can also be a little bit plastic and might be related to environmental factors. And then sometimes there's sexual dimorphism. So for instance, female stoneflies tend to be larger than males, and so they may go through more instar stages than males. So in HEMIMETABOLOUS metamorphosis, the final instar nymph transforms directly into an adult. And in this case, it loses a lot of its features like gills. And for mayflies, as shown in this picture, it actually goes through two winged stages. The first is called the subimago, or the dun, And when it emerges, it's a little bit dull colored. It's very hairy, and that's to escape the water. So it has to emerge underwater. And so the subimago is hydrofuge, and so it resists getting wet, and that allows it to break through the surface of the water. And then it goes through a second stage. It will shed its old exoskeleton and emerge again as an imago or spinner, which is the sexually mature form. You can see has much longer cerci and forelegs. And some of them actually lose some of their other legs in this imago stage.